Development of brinchial or pharyngeal arches and the primitive mouth. So when the stomodium first forms, it is limited by the frontal prominence rostrally and caudally by the developing cardiac bulge. The backopharyngeal membrane, which is a bilaminar structure, consists of a post-ectoderm and endoderm. This backopharyngeal membrane separates the stomodium from the foregut. So this soon breaks down so that the stomodium communicates directly with the foregut. So laterally, the stomodium is limited by the first pair of pharyngeal or brinchial arches. So as a result of proliferating lateral place mesoderm and subsequent reinforcement by migrating neural crest cells, brinchial arches form in the pharyngeal wall and result in 6 cylindrical thickness. So note that arches 5 and 6 are transient structures in humans and are not visible on the surface of the embryo. This thickening expands from the lateral wall of the pharynx, pass beneath the flow of the pharynx, and approach their anatomic counterpart, which is expanding from the opposite side. Hence, the arches progressively separate the primitives to the stomodium from the developing heart. The arches are seen as bulges on the lateral aspect of the embryo and are separated externally by small clefts called the brinchial grooves. On the inner aspect of the pharyngeal wall, we see corresponding small depressions called the pharyngeal pouches. It separates each the brinchial arches internally. Each brinchial arches has the same basic plan. The inner aspect is covered by endoderm, but in the case of the first arch, the inner lining is ectoderm because it forms in front of the backopharyngeal membrane. So the outer surface is covered by ectoderm. The central core consists of mesenchyme derived from the lateral plate mesoderm. This area is invaded by mesoderm from the neural crest, referred to as ectomesenchyme. This ectomesenchyme condenses to form a bar of cartilage, referred to as the arch cartilage. So the cartilage of the first arch is called Meckel's cartilage, and that of the second arch is called the Reischer's cartilage. And time surrounding the first and second arch cartilages gives rise to the muscles of mastications and to the muscles of facial expressions, respectively. Each arch also contains an artery and a nerve. So the first, second, and third brinchial arches play an important role in the development of the face, mouth, and the tongue. So the first, second, and third brinchial arches play an important role in the development of the face, mouth, and the tongue. So the formation of the face is described in terms of the formation and merging of several processes and prominences. So the term merging is described as swelling of processes with the elimination of a furrow between them. So fusion, on the other hand, occurs in the union of the palatal processes. So such fusion involves the breakdown of surface epithelium. So the primitive stomodium is at first bound by the frontal process rostrally and caudally by the developing heart, and laterally by the first brinchial arch. So with spread of the arches mid-ventrally, the cardiac plate is eliminated from the stomodium and the flow of the mouth is formed by the epithelium. And time of the first, second, and third brinchial arches. At about 24 days, the first brinchial arch establishes another process, the maxillary process. Now, this stomodium is limited cranially by the frontal prominence, which covers the rapidly expanding forebrain, laterally by the newly formed maxillary process, and eventually by the first arch, which now becomes known as the mandibular process. So early development of the face is dominated by the proliferation and migration of ectomesenchyme involved in the formation of the primitive nasal cavities. At about 28 days, localized thickness developed within the ectoderm of the frontal process, thus rostro to the opening of the stomogium. So this thickening are referred to as olfactory placodes. Rapid proliferation of the underlying mesenchyme around these olfactory placodes results in bulges of the frontal eminence and also produces a horseshoe-shaped ridge that converts the olfactory placodes into nasal pits. So the lateral aspect of the horseshoe is called the lateral nasal process, and the medial aspect is the medial nasal process. So the region of the frontal prominence where the nose will develop is referred to as the frontonasal process. So the medial nasal process of both sides, together with the frontonasal process, gives rise to the middle portion of the nose, the middle portion of the upper lip, and the anterior portion of the maxilla, as well as the primary palate. So the maxillary process grows medially and approaches the lateral and medial nasal processes, but remains separated from them by two distinct grooves: the nasal optic groove and the bucconasal groove. So the medial growth of the maxillary process pushes the medial nasal process towards the midline, where it then merges with its anatomic counterpart from the orifice beside the upper lip. Is therefore termed from the maxillary process of each side and the medial nasal process. The lower lip is formed by merging of two streams of ectomesenchyme of the mandibular process. So note, the merging of the two medial nasal process results in the formation of that part of the maxilla that carries the incisor teeth and the primary palate, and also a part of the lip. So, at about 30 days of gestation, the epithelium of the inferior border of the maxillary process and the superior border of the mandibular arch begins to proliferate and thicken. So, this thickened area is referred to as the odontogenic epithelium. At this time, the primary epithelial band can also be observed. So, the primary epithelial band is a single plate of thickened epithelium, described as an arch-shaped, continuous plate of odontogenic epithelium. So in the upper jaw, it forms four separate zones of the epithelial proliferation. So that is laterally, one zone associated with each maxillary process, and in the middle one, with each medial nasal process. So in the lower jaw, it forms from two zones. That is one from each mandibular processes. So at this time, we have a common or own nasal cavity. This is bound anteriorly by the primary palate, and the cavity is occupied mainly by the developing tongue. Only after the development of the secondary palate does one get the distinction between the oral and the nasal cavities. So the palate proper develops from the primary and secondary components. The formation of the secondary palate commences between 7 and 8 weeks and completes around the 3rd month of gestation. So 3 outgrowths appear in the oral cavity. The nasal septum grows downward from the frontonasal process along the midline, and two palatine shelves, one from each side, extend from the maxillary processes towards the midline. The first, the shells are directed downward of each side, of the tongue after the 7th week. So the head of the embryo tilts back. The tongue is withdrawn from between the shelves, and these shelves then elevate and fuse with each other above the tongue and anteriorly with the primary palate. So the septum and the two shelves converge, fuse along the midline, and hence separate the primary or the nasal cavity into nasal and oral cavities. So the tongue begins to develop at about 4 weeks. The pharyngeal arches meet in the midline beneath the primitive mouth. So local proliferation of ectomesenchyme then gives rise to a number of swellings. The floor of the mouth. The first swelling arises in the midline of the mandibular process. So this is referred to as the tuberculum impar. So this is then flanked by two other bulges referred to as lingual swellings. So these lateral swellings enlarge and merge with each other and with the tuberculum impar to form a large mass from which the mucous membrane of the anterior two-thirds of the tongue is formed. So the posterior third of the tongue arises from a large midline swelling developed from the mesenchyme of the second, third, and fourth arches. So this swelling consists of the copula, which is associated with the second arch, and the large hypobranchial eminence. It is associated with third and fourth branchial arches. So as the tongue develops, the hypobranchial eminence overgrows the copula, which then subsequently disappears. So the posterior part of the fourth arch, it marks the development of the epiglottis. So the tongue separates from the floor of the mouth by a downgrowth of ectoderm around its periphery, which then subsequently degenerates to form the lingual sulcus. So this allows mobility of the tongue. The muscles of the tongue have a different origin they arise from. The occipital somites, which have migrated forward to the tongue area, carrying with them their nerve supply, and the 12 cranial nerve or the hypobranchial nerve. Development of the nose. So the nose is formed from the 5 facial prominences. As I've said earlier, the 5 facial prominences are what you call the frontonasal prominence, 2 maxillary prominence, and the 2 mandibular prominence. So the frontal prominence gives rise to the bridge and nasal septum. So the medial nasal prominences provide the crest and the tip of the nose, and the lateral prominence side, and the lateral nasal prominence is forming the side or the alley of the nose. So the olfactory pit forms the nostril and then becomes deeper to form the blind sac or what we call the vestibule. Here. The nasal cavities are separated from the mouth by the development of the palate. So the narrowing of the frontonasal process and the enlargement of the nasal cavities brings them closer together. So the intervening tissues becomes much thin from the nasal septum. And also the nasal pit now deepen to form the nasal sac, which will then expand both dorsally and caudally. So in this diagram of the face, the broken lines indicate the possible location of the facial cleft. So here are the broken lines. These are the possible sites for the cleft. And the areas between the broken lines correspond with the areas formed by the original facial processes. So shown here is a diagram of the hard and soft palate as they appear viewed from below. So the area of the front teeth corresponds with the primitive palate. In this part, the primitive palate, formed largely by the 2 medial nasal process. So when the 2 medial process emerge, it will form the primary palate. So the shaded part of the palate, in this part, so the palate has 2 parts, the hard palate in the front and the soft palate in the back. Hard palate and the soft palate in this part. So it is formed by the 2 palatal processes. which come together and fuse in the midline. So clefts are possible between the nasal process and the palatal process, and in the midline between the two palatal prominence or the processes. So the facial malformations arises from week 5 to week 12. So developmental malformation or cleft. Facial and palatal clefts usually occur due to a combination of environmental and hereditary factors. So the cleft lip is the most common type of the facial malformation. So the most critical period for the development of the palate is from the end of week 6 to the beginning of the week 9, where abnormalities like cleft palate can develop. So now let us go to the cleft lip. So a partial or complete failure of maxillary process to fuse with the median nasal process results in a cleft lip. So a cleft may affect one or both sides of the lip. So it is unilateral or bilateral, and it can be incomplete or complete. So shown here in this picture are the types of the facial cleft. First, the normal one. Letter B, the unilateral cleft lip in this side. And letter C, the bilateral cleft lip, because 2. Then here, the median cleft lip, here, or what we call the hair lip. Letter E, oblique facial cleft. And letter F, the median cleft or frontonasal dysplasia. Letter G, the lateral facial cleft. Here, lateral, because it is located on the lateral side. And letter H, the mandibular cleft, since it originates from the mandible. On the other hand, these are the palatal clefts seen from the ventral view. So the first, the normal one. Next, cleft lip and the alveolus, because it is involved. The alveolar bone is involved. Then letter C, cleft of lip and primary palate. The unilateral cleft lip and the palate. Next, bilateral cleft lip and primary palate. Next, the bilateral cleft lip and the palate. And the last is the cleft palate only, because it is located only in the palate region. So the unilateral cleft lip involves the deformity of the lip in addition to the alveolus and the nose. So treatment goals include the restoration of the facial appearance and oral function, improvement of dental, skeletal, and occlusal relationships, improvement of speech, and the psychosocial space. So next is the unilateral complete cleft lip. So in this condition, the maxillary and the medial nasal processes did not fuse and then pulled during the facial growth. So the cleft may extend from the lip up through the floor of the nasal area, a complete unilateral cleft and alveolus, or it may affect the dental ridge and the lip itself. So the next one is the bilateral complete cleft of the lip. So in this case, this is the clinical view of the bilateral complete cleft lip. So the medial labial process did not fuse on either side of the developing maxillary process. So this results in anterior extrusion of the medial labial process. So if the cleft lip is bilateral, it's likely that the palate also has a cleft. So although bilateral clefts can be symmetrical, the appearance on both sides of the mouth isn't always the same. So it may be asymmetrical, so perhaps with a groove on one side of the mouth and a cleft on the other. So median cleft lip is a rare anomaly consisting of a midline vertical cleft through the upper lip. So it can also involve the premaxillary bone, the nasal septum, and the central nervous system. The next one is the mandibular cleft. So the two parts of the first pharyngeal arch, which is called the mandibular arch, including the bony mandible, are separated at birth in this rare condition. So these again are the examples of the cleft lip and the palate. The first one has cleft lip only in this side. Next, the cleft palate only in this part in the palate region. Letter C, the unilateral complete cleft lip here, because only one. Then um, the bilateral complete cleft lip, and the palate in this side. So, thyroglossal cyst. A thyroglossal cyst is a fibrosis that forms persistent to thyroglossal duct. So, a thyroglossal cyst is a fibrosis that forms from a persistent thyroglossal duct. So, thyroglossal cyst can be defined as an irregular neck mass or a lump which develops from cells and tissues left over after the formation of the thyroid gland during developmental stages. So, failure of complete obliteration of the cervical sinus results in cervical cyst or brachial fistula. So, they may be found on the neck along the anterior border of the sternocleidomastoid in this part. Here, this is where the obliteration can be seen. So, the next condition is what we call the Treacher-Collins syndrome, or what we call the mandibulofacial dysostosis. So, in this condition, abnormal development of the structures from first and second pharyngeal arches, and it is autosomal dominant. So, next is the Pierre-Robin syndrome. So, in this condition, the patient has cleft palate, retrognathia, macroglossia, and acloglossia uncommon, and glossoptosis and micrognathia. Then, so the Pierre-Robin sequence or syndrome is a condition in which an infant has a smaller than normal lower jaw. So, a tongue that falls back in the throat and difficulty breathing. So, it's present already in birth. So, a Treacher-Collins syndrome, TCS, is an autosomal dominant congenital disorder characterized by craniofacial deformities. So, typically, it involves the ears, the eyes, the cheekbones, and the jawbones. So, those affected have normal intelligence naman. So, in terms of genetics, including inheritance patterns and frequency. So, it is autosomal dominant, as I've said earlier. Then, inheritance pattern. So, a small percentage has autosomal recessive inheritance. So, the clinical feature of Treacher-Collins syndrome or TCS consists of bilaterally symmetric, abnormal development of the structures arising from the first and second brachial arches. So, this abnormal development contributes to deficient generation of the lower two-thirds of the face. What's amazing about tooth development is how early it starts. So this embryo is only 6 weeks old, and yet the epithelium over its mandible is already thickening in preparation for what's about to happen as it grows thicker. So the epithelium creates an island of cells which is the origin of the new tooth. So this is the dental lamina, and its remarkable potential is starting to unfold as we get closer. So we can see other key players in the formation of our dentition. So Meckel's cartilage provides support for the developing tissues and the blood vessels that help shape the embryonic progress. By now, the lamina has grown into a bud. The epithelial cells haven't changed much, but as the bud grows, it starts to bend into a dome in response to pressure from the blood vessels and the mesenchymal cells underneath condense. At this point, we can already identify the cells that will form the tissues of the mature tooth. So the enamel organ is made of cells from the epithelial cap, and it will generate the enamel of the tooth. So the condensed mesenchymal cells will form the dentine and the pulp, and they are called the dental papilla. So the dental follicle will develop from the remaining condensed mesenchyme, and all of these cells together as known as the tooth. Tooth germ so far. So we've seen the bud and cap. So the third and most dramatic is the bell stage. As the tooth germ begins to grow, the cells differentiate. The stellate reticulum is now obvious. This is made up of star-shaped cells pushed apart by fluid pressure, and it separates the inner and outer enamel epithelium. Just visible is a flattened area of cells called the stratum intermedium. So we can also see the junction where the inner and outer enamel epithelium meet. So this cervical loop is important in tooth formation, as we'll discover soon. By the time the tooth bud pinches off from the dental lamina at the end of the bell stage, the inner enamel epithelium has folded to form the shape of the future crown with a scaffold to work on. So next is the salivary gland development. So the parotid and the submandibular salivary glands appear in the connective tissues of the developing cheek in the 6th week. Next, the sublingual glands develop at around the 8th week. So let us now look at the development of the salivary glands. Three major pairs of salivary glands originate uniformly as oral epithelial buds involving the underlying mesenchyme. So all the parenchymal or secretory tissue of the glands arises from the proliferation of the oral epithelium, which is either ectodermal or endodermal in origin. So the stroma, or what we call the capsule and the septa of the gland, originates from the mesenchyme that may be of either mesodermal or neural crest origin. So lastly, the failure of the bud to canalize to form duct before acinar cells start secretion, covering cells results in retention types of teeth.
The cranium is where the brain or the cranium is given supported by the cartilages. The development of the chondrocranium anteriorly is here the nasal capsule. More posteriorly is the pituitary, and most posteriorly is the occipital cartilages, and laterally is the otic capsule. Second is the development of the maxillary complex. Here in letter A, the nasal capsule represents the maxillary skeleton. Here is the metals cartilage that represents the mandibular skeleton. Here in letter B is the developing of the body of the mandible at endocondyle at 16 weeks. Here in letter C, the coronoid process is evident as the appearance of the temporomandibular joint. Here in letter D, at 30 weeks, the membrane bones of the nasomaxillary complex develop externally to the endochondral bones, replacing the cartilage of the midline of the bone. Third is the development of the bony palate. Let's go to 8 weeks. During the 8 weeks, the shelves are horizontal and grow towards the midline. Anterior palate gives rise to the premaxilla and the maxilla. In the 9 weeks, the shelves are in near contact here, and the premaxillary maxillary ossification centers appear. During the 10th weeks, the soft tissue of the palate has fused and ossification centers of the premaxilla maxillary grow medially. Here. And in the 14th weeks, the premaxillary bone supports the incisors, maxillary bone supports the cuspids and the first molars, and the palatine bone supports the second molars. Fourth is the development of the mandible and the temporomandibular joint. The primary cartilage is the Meckel's cartilage, where is the rod-shaped bar in the lower part of the face. It is derived from the first brachial arch. Here, in this posterior area, the incus and the malleus becomes the bones of the middle ear. The incus and the malleus function to provide a movable joint mandibular condyle develops in relation to the glenoid fossa of the temporal bone. The secondary cartilages are the condylar cartilage, which will give rise to the condyle, the coronoid cartilage, that give rise to the coronoid cartilage, and the symphysial cartilage here that will give rise to the mental ossicle. The facial muscles is where the superficial layer give rise to the platysma muscle over the mandible to the cheek, forehead, and the temporal region. While the deep layer gives rise to the anterior, superior, postauricular frontalis and occipitalis muscles. Overlying the vaccinator muscle is the buccal fat pad here that develops extending between the masseter and the temporalis muscle. These functions causes the cheek to appear plump in newborn. The masseter are the muscles of mastication. The muscles of mastication are the lateral pterygoid muscle, the medial pterygoid muscle, temporalis muscle, and the masseter. The masseter muscle begins attachment to the zygomatic arch, providing space for the muscle development. Now let's go to the fetal period from 3rd to 9th month. First is the craniofacial changes. The craniofacial changes is the face assumes more human appearance. The eyes are now directed forward and the eyelids have grown together and are fused. The head is erect and the bridge of the nose becomes somewhat prominent. As the face grows downward and forward, as the face grows downward and forward, the ears appear on a horizontal plane with the eyes instead at the lower corners of the face. The head increases in length, width, and height proportionally. Next is the radiographic changes. This is the premaxilla, the maxillary, and the palatine. In radiographic changes, the palate increases in length 4-fold. The maxillary region increases 5-fold. First signs of cranial base bone appear, and the sphenoethmoidal and the spheno-occipital synchondroses are evident.